mini talk. And today we are very happy to invite Chang Jinwu, who comes from the University of Hong Kong. He is working with Professor Jingyao Tang, and his talk will be on this topic, an exchange-enabled synthetic swarm, which was recently published in Nature Nanotechnology. So without further ado, I'll pass the stage to Changjin. Please go ahead, Changjin.
Okay. Thank you, Chenxi, for the very nice introduction. Okay. Hello, everyone. This is Changjin Wu from Hong Kong, China, and I'm a PhD student in the chemistry department at Hong Kong University, and I'm doing research following Professor Jingyao Tang in the nanomaterials research group. So today, it's very happy to share with you our previously published work, so exchange-enabled synthetic swarm. So basically, in this work, we observe the very interesting collective dynamics in the artificial active matter system that resembles those intelligent collective behavior in the biological systems. So these two videos here, the left one is the flocking of birds in nature, and the right one is our system. Okay, so before starting my presentation, there are some people I want to acknowledge. So first, I want to thank my supervisor, Professor Jingyao Tang, for his continuous support and instruction in this project. And I also want to thank Professor Mingcheng Yang for his help with the simulation. And Professor Mingcheng Yang is from the Institute of Physics in the Chinese Academy of Science. And as people in the authorship, they all have given me a lot of help in preparing the paper. So now let's move into the demonstration of the work in this paper. So first, I will quickly go through some background of this work. So our system, we think that it's a model system of the complex system. So in the complex system, it normally it is composed of a large number of the decentralized subsystems, which can self-organize where different control structures into the complicated emergence through the hierarchical development. And one example of the complex system is collective intelligence in nature. As in these videos, so the flocking of birds, the schooling of fish, and the ant army, and the crowd of human. So if you consider the length scale and the intelligent level of those individuals in these systems, they are clearly different, but they still demonstrate a similar collective behavior. And in these systems, those individuals can self-organize from the very disordered and chaotic state into the very ordered state. So that's why we also call the collective intelligence the edge of chaos. So the collective intelligence in nature, they are so fascinating, but however, they are also very complicated. And the scientists from different fields, like those are doing with chemistry, materials, and engineering, just like us, we are also want to reproduce those collective dynamics in the biological systems, in the synthetic systems. But this is very important. Like this year, the Science Journal pointed out 125 most important questions for nowadays research. And there are two of them. One is that will the injectable disease-fighting nanorobots ever be a reality? And another one is how does the global intelligence emerge? So if you combine these two questions, you get another one. It's actually about the artificial collective intelligence in the nanorobotic system. So so today's nanorobots, especially those synthetic nanorobots, they're actually something about the nanoscale synthetic active particles. Those active particles can collect the energy from the environment and convert it into the mechanical motion. So so they are operating far from the thermodynamic equilibrium state. So in the population level, those active particles can align their moving direction with the average moving direction of the whole members in the community. So together they can show the very self-organized collective motion behavior, where compared with the active particles, they are passive particles, which are always at the thermodynamic equilibrium state. And even in the populus level, those passive particles, they are still isotropic for each passive particle. So in this case, if you calculate the mean velocity of those passive particles in the system, that will always be 0. So if you're doing with the active particle suspension, you probably will observe the phase transition or collective motion of those particles, like in this work. Their particles can be activated by illuminating light on the particles. And once those particles are activated from the passive to active, they can interact with each other, with the surrounding particles, and gradually they can demonstrate those collective behavior. So we can understand this the collective behavior of the active particles by using the potential energy landscape here. You consider that you have active particles in one configuration, and it is self-assembled collective behavior is in another configuration. And there is some energy fluctuation in the environment. So when you import energy for the active particles, those active particles can absorb the energy and they can transit. If the energy is larger than the barriers between the two configurations, those active particles can transit from one configuration into another. So if you go back to those collective intelligence in nature and compared with collective behavior in the synthetic systems, you will find that there is always information flow and feedback between those active individuals. So those individuals can coordinate their behavior and gradually demonstrate the self-organized collective behavior. So this interaction is actually the non-reciprocal interaction. And if you study study different active systems, you will find that the non-reciprocity always exists in those active systems. That's because the non-reciprocity can continuously break the symmetry of the system and maintain the system as a dynamic and the non-equilibrium state. So this inspires us that if we want to reproduce those collective dynamics in the biological systems, we need to in the synthetic system, we will need to construct the non-reciprocal interaction between those active matters. So what we actually did is that we are inspired by the symposium system in nature, and we synthesized the micro and nanoscale propionate active particles, and then we construct the non-reciprocal reaction between those active particles. So the active particles we used are zinc oxide rod and sulfonated polystyrene microbeads. Like we first synthesized the zinc oxide rod on the silicon wafer, and when we transfer those nanorods into water, we observe that those nanorods can swing. That's because zinc oxide rod it has a different surface potential at different phases. So when you put those nanorods in water, the dissolution rate at the different phases are different. So they can propel themselves by the self-diffusive process. And since the propulsion is due to the dissolution and the self-diffusive process, so we can detect the zinc ions released from the in the solution from the nanorods, and the propulsion is dependent on the solution pH. And another active particle we used is a sulfonated polystyrene microbeads, and it is we also call it SPS particle, and it is a cation exchanger, and it has been reported before our papers that the exchangers in the solution can generate the electroosmotic flow around the exchanger particles due to the exchange reaction with the ions in the solution. So that osmotic flow can gradually self-assemble those particles together in the solution. And in our work, we also synthesized the very small sized SPS particles, and we put them in water. We observe the self-assembly behavior. Now, when we mix the nanorods and the SPS particles in water, we observe the very interesting interaction between these two particles. as in this video, the attraction, repulsion, and assemble behavior interaction. And this interaction is due to the ion-exchange reaction between the zinc oxide rod and the SPS particles. So during the motion of the zinc oxide rod, it will release the zinc ions in the solution, which can be absorbed by the SPS particles. So the SPS particles will produce the proteins in the solution, which will in turn consumed by the zinc oxide rod. So this ion exchange reaction couples these two kinds of particles and enhance their reactivity and proportion simultaneously. So because the zinc oxide rod can generate the zinc ion gradient, so that will induce the outward electroosmotic flow around the glass substrate. So in the video, we can see that the particles around the nanorods is pushed away from the nanorods. So this speed pulse here indicates the assemble state of the particles. When they are approaching each other, those reactivity activities are being enhanced simultaneously, so we can see the speed increase. Okay, when we are looking carefully into the repulsion repulsion interaction between the nanorod and SPS particles, we found that when the SPS particles is approaching the nanorod, its speed is increasing with the separation distance decrease. While when they are leaving from the zinc oxide rod, their speed is decreased with the separation distance increase. So this interaction between the zinc oxide rod and SPS particles is actually a separation dependent communication, and this is very important in our system, and it will induce the swarming behavior in our system, which I will introduce later. So this interaction between the zinc oxide rod and SPS particles is actually in 3 dimensions. Like when we fix the zinc oxide rod on the glass substrate, we observe that those the SPS particles will be attracted to the nanorods from the upper solution, and then they are pushed away around the glass substrate from the zinc oxide rod. And at the same time, we observe that when the SPS particles are approaching the zinc oxide rod, their speed is increasing, and when they're leaving from the zinc oxide rod, their speed is decreasing. So this 3D velocity profile also shows the separation distance regulated communication between these two kinds of particles. And because the interaction between the zinc oxide rod and SPS particles is due to the ion exchange reactions, so we can use the pH probe to map the pH profile of the particle system. So since during the interaction, the SPS particles will produce a protein gradient, so we can see a lower pH region near the SPS particle. And since the zinc oxide rod can consume the proteins, so near the zinc oxide rod, we can see a higher pH region. And we think that as long as there is an exchange reaction, we can see that we can always see the repulsion attraction between particles. So here we replace the SPS particles with a morph particle, and we replace the zinc oxide rod with a calcium carbonate nanocube. So we still observe the similar attraction repulsion interaction in the binary active particle systems. So here we used the console simulation to demonstrate to integrate the attraction repulsion between the zinc oxide rod and the SPS particles, and also the cluster formation process in our system. So during the motion of the zinc oxide rod, it will generate the zinc ion gradient that will induce the outward electroosmotic flow, and during the interaction of SPS particles, it will generate the inward attractive osmotic flow. So in this video, we can see that around the zinc oxide rod, particles will be pushed away from the zinc oxide rod, and in a distance away from the zinc oxide rod, those SPS particles can self-assemble with each other. And with the increase of the particle number of the SPS particles, they can form a coherent structure like cluster with a zinc oxide rod in the center and SPS particles at the perimeter. And this cluster formation process is also originated from the ion exchange reaction. So we can also use the page probe to map the page profile of the cluster. So in the center of the cluster, there is a zinc ion gradient. So we can see a pair page region in the center, and as perimeter, there are SPS particles aggregation. So we can see, the lower page region in that part. Okay. We also studied the swarming behavior. We later investigated swarming behavior in our active particle system. Like I introduced in the background, that the active particles can always, demonstrate the collective behavior in the populace level. And in our system, the ion exchange reaction can fetch the very strong osmotic interaction and separation dependent reactivity. So we were expecting to see, some more interesting collective dynamics in our system. And indeed, we observed the very non-trivial collective behavior in the zinc oxide SPS active particle system. As in these videos, in the macro scale, those particles can form the coherent structure like a cluster with a zinc oxide lateral in the center, and that is surrounded by the SPS particles. And in the larger scale, we can those clusters they are very dynamic and active, so they're merging and splitting. Well, in the even larger scale, we observe the alignment and cohesion collective behavior. Those small clusters can gradually form a very large cluster. So we adopted the coarse-grained model to simulate the swarming behavior in our system. Professor Minton Yang helped us with the simulation. So in the simulation, we consider three basic interactions, like the excluded volume interaction and the repulsive osmosis from the zinc oxide rod and the attractive osmosis from the SPS particles. So the first video shows the simulation result of our system. That's a very strong swarming behavior. Just like I saw it in the, I mentioned in the previous slides, that the separation dependent reactivity is very, very important to induce the swarming behavior in our system. So when we remove the, the separation dependent reactivity in the simulation, the swarming behavior is severely weakened. Those particles can only form the very small clusters. And when we remove osmosis interaction between those particles, because those particles can no longer interact with each other, so they cannot show the swarming behavior anymore. Okay, we also studied the phase transition behavior of our particles in the confinement. So we first can consider a simple situation that we have the passive particles in the confinement. So for the passive particles, they have their distribution in the confinement satisfy the Boltzmann distribution, like in this equation. Like for passive particles, they have the constant energy potential, so they will have the very uniform distribution in the confinement. Like in this video here, we put the passive PS particles in the glass patch dish. So we observe that they will eventually uniformly distribute in the patch dish. However, if you are doing with active particles, because those active particles, they can interact with the confinement wall and they can interact with each other. So there's a confinement wall, there will be the fair probability of those particles to present. So for active particles, their distribution in the confinement wall is a non-Boltzmann distribution. And for our system, we put the mixture of the zinc oxide rod and SPS particles in the glass petri dish, and we observe the very interesting center-verge phase transition collective behavior. Like in this video, our particles, just like normal active particles, they can aggregate as a confinement wall. However, in our system, those particles can show the very dynamic clustering and swarming behavior, and they can so and those small clusters can contract into very large clusters. So in the center, we still we also observe a very large cluster. So we finally observe this center-verge phase transition behavior, and we also used the applies cost gradient model to simulate the center-verge phase transition behavior of our particles in the simulation. And this one is not our experimental data. I just draw this curve to demonstrate the the non-Boltzmann distribution of the target particles in our system, and it is center-verge phase transition behavior. Okay, so the phase transition behavior in our system is quite similar to the carbon sensing behavior in the biological systems, like in the microorganism systems. So for microorganism, like a bacteria, in the low density, those bacteria can secrete the kind of chemical signal like autoinducer to communicate with each other. While in the high density, those the concentration of the autoinducer increase, so the communication between those bacteria also increase. In that situation, those bacteria can make consensus decision to emit light. So this quorum sensing behavior is actually a population density regulated collective behavior. And this quorum sensing rule has also been adopted to understand the phase transition behavior in the active particle systems. So we also tested the quorum sensing behavior in our system. So we put the mixture particle in the irregular containers. So in these two containers, they both have a corner that has the largest curvature compared to other areas in the container. So in that corner with the largest curvature, they can confine the particles better in the very beginning. So the attractive osmosis that parties that corner is larger than other area in the container. So it can attract more and more particles to aggregate in that container in that corner. So eventually all the particles will migrate to that corner. So the quorum sensing behavior in the biological system is actually the consensus decision-making process. Like in this work, they put some fish in the water pool, and they have two targets at a large end of the water pool. So they just put the fish in the pool and let them go. And the fun side, those fish will collectively swim to one target in this water pool, rather than splitting the groups into two smaller groups towards two targets. This is very interesting because in the biological systems, those living matter always tend to maintain a large group, because a large group is better for the community to survive and adaptate into the environment. Well, for our system, we also put the mixture in this irregular container, and for this container, it has two corners that has the same largest curvature. So you may expect that those particles will eventually evenly distribute as two corners, but they didn't. They actually run together to collect to one corner in this container. So this is very interesting and very important that we finally synthesize a system that is so close to the intelligent collective behavior in the biological systems. So just like those found in fish and honeybees, and we can find some very fundamental rules in between the collective intelligence in the biological systems and the collective dynamics in the synthetic systems. That's just like I mentioned in the background, that they all have the information flow and the feedback between those active individuals. And those individuals can coordinate their behavior, and then together they can self-organize to demonstrate the collective motion behavior. And And then we also did some control experiments. We added some EDTA in the solution. Because those EDTA can combine the ions in the solution, so they can block the ion exchange reaction. So when the ion exchange reaction is weakened, so the swarming behavior, you can see in this video, the swarming behavior almost disappear. And then we also added a SPS aggregate in the glass patch dish because SPS particles can generate the attractive osmosis, so they can attract the particles away from SPS aggregate. So finally, all the particles in the gas patch dish will collectively swarm to that SPS aggregate. So here I want to have an outlook of our mechanism, like what we can do with the current decision-making mechanism of active particles. Below that, the nanorobots have been envisioned as drug delivery carriers for the precision medicine for a very long time. And imagine there's a kind of disease in the human body, so we can inject the nanorobots inside the body, and those nanorobots can deliver the drug to the disease directly by the chemotaxis. However, this is very, very difficult and almost impossible because the nanorobot, because we know that the chemotaxis is actually a short-range effect, normally within 1 millimeter or several hundred micrometer range. So that means that those nanorobots, they cannot even sense the disease and cannot do the non-distance drug delivery. So however, for our system, our active particles can show the macro scale collective motion behavior without the the dependence of the short range chemotaxis. So we believe that it will be permissive for the non-distance drug delivery. And this is actually what we are doing right now in our lab. And we can also find the quantization mechanism in the biological systems, like the neutrophils, they can collectively swarm to the injury by the chromatin rule. So here I want to post at the end, I want to post an advertisement for our department. So our department always welcomes the young students want to do postgraduate study in our department. So the research area in our department is quite broad, and we have different labs in doing research in different fields, like in the chemical biology, and material science, and energy, and synthetic chemistry, including the inorganic and the organic chemistry. And we also have the labs in the physical chemistry and computation. So if you are interested, you can go to open our department website and take a look and get more information. And yeah, we also have a Q&A session next Thursday on Zoom. And in Zoom meeting, our department head Professor Chi will introduce our department, and Professor Chi will probably answer questions. Okay. I probably will stop my presentation right here. So I will be happy to take any other question.
Okay. Thank you, Changjin. Thank you for your inspiring and wonderful talk, which is really impressive. So next, we are moving on the Q&A session. Let's say if we have some question from audience.
Yeah.
Please feel free to just unmute yourself to ask these questions.
Hello? Hi, can you hear me?
Yes, I can hear you.
Yeah. Hi, Changjin. First of all, thank you very much for your wonderful talk. It's indeed a very inspiring topic. One question though, I saw that video on slide 7. Can you go to the slide 7?
You mean page 7, right?
Yeah.
Okay, here.
Yeah. I was wondering, why do some zinc oxide nanorods have the rotational trajectories while others are just moving directionally?
Okay, okay. Thank you for your question, and it's a very good question. And so the proportion of the zinc oxide rod is due to the the surface potential difference at the different phases. So like at the polar zinc phase, it will have the higher surface potential. So the dissolution rate at the polar zinc phase will be higher than other phases. So it's reasonable to say that when you put the rod in the water, those nether rod can propel themselves around the non-axis of the nether rod. However, when we prepare the zinc oxide nether rods, there are always some defects on the set surface of those nether rods. So those defects can offer the additional dissolution side. So they can, during the propulsion, they can offer the rotational torque of the propulsion. So they can rotate the nether rod. That's why we can say some nether rod goes the linear trajectory, and some nether rods have the very rotational trajectory. Yeah.
Thank you very much.
Okay, thank you.
So is there any more question from audience? We got a question from the chat box. Sewan asked, why is the pH profile only shows a range from 5 to 8?
Okay, thank you for your question. That's because the pH probe, we use is can only detect pH range from 5 to 8, and of course you can, if you use other pH probe, maybe you can do like a more accurate pH mapping for the pH profile in the particle system. However, this is not, we do not really care about the real value of the pH near the particles. We care more about the pH gradient near the particles. We want to know during the ion exchange reaction, there should be lower pH region near the SPS particles, and there should be a higher pH region near the zinc oxide rods, and this is most important thing we want to know to verify that there's indeed those interactions due to the ion exchange reaction. Yeah.
Okay, thank you for your answer.
Thank you.
So I have a question, Tangjin. For the swarming behavior, so why this nanorods didn't push away the SPS particles?
Oh, okay, yeah. That's a very good question. Well, just like this video. So in the very beginning, you can imagine that those because the zinc oxide rod they can generate the automatic electro-osmotic flow, so they can push the particles away from the nether rod. However, in a distance away from the nether rod, those automatic electro-osmotic flow is weakened, right? Because that electro-osmotic flow is dependent on the distance, separation distance. So at this distance, those the SPS particles, they will also generate the attractive osmosis. So in that distance, the outward electro-osmotic flow and the attractive osmotic flow, they are balanced. So those particles, they can aggregate, they can self-assemble. And when the particle number of this SPS particle increase, they can generate stronger and stronger attractive osmosis. So you will see more and more particles aggregate around the zinc oxide rod. So they can form the cluster with the zinc oxide rod in the center and the SPS particles as a perimeter. They are together, is very like a partial structure cluster. Yeah.
Okay, fantastic. Thank you for your answer. We got another question from the chat box.
Yeah, so the system do not show this behavior in the bulk, but only close to the surface. I think you're meaning that those, the attraction, repulsion, the interaction is near the glass substrate, right? Yeah, you're very correct that we, when we are observing those behaviors, we always focus our, microscope at the glass substrate. And that's because, for the outward electro-osmotic flow, that's, that osmotic flow is due to the, along the glass substrate. So in the bulk of the solution, of course, there is some like electrophoresis or chemotaxis behavior, we can say, but it's weaker than the electro-osmotic flow near the substrate. So maybe we can say some, there is some, similar protection repulsion behavior, but it should be much weaker than those near the glass substrate.